Okay, I'm going to go ahead and get started. Uh, just to give you a quick overview of where we're at, I know I've been running long every time, and so I decided to punt on the EQTL analysis for today. I gave myself 4 lecture periods to do machine learning, and so we'll basically just be cutting that by 1 lecture period so that we can do sort of the marriage between genome variant analysis from the last module with the transcriptome stuff that we're doing this week. So seeing how those two come together. That also gives me a chance to spend some time on why multiple testing corrections are important, which I mentioned briefly on Tuesday, but is a pretty significant issue for bioinformatics, just because you end up working on big data sets, doing lots of tests. Right? We talked about if you do 1 million SNPs, you're going to get 50,000 false positives at 0.05, even if there's no associations. So today's assignment, or this week's assignment, will just be to finish the Colab notebook. And just like last week, just upload the figures that get generated at the end of the Colab notebook. So to that end, the Colab notebook link is there. Go ahead and do that. One of the steps takes a few minutes, so we're going to go ahead and start with the Colab notebook, and then I'll go back and complete the notes while one of those cells runs. And it's not even an interesting cell, it's just an install step. So go ahead and get that notebook. Let me go to it. So when you get there, it should look something like this. And first step should look pretty similar, because QC and error correction is the same for RNA-Seq as it was for genome variant analysis. So this FAST-P step, installing that software, should go pretty fast. If the bandwidth can support you all. The Wi-Fi bandwidth, that is. So yeah, first thing is go get the Colab notebook on the Canvas link. We're just going to start the first few cells because one of the install steps takes a while. Right. So second step is to simulate a transcriptome. So we're just going to use simulated data, and I'll show you after we start running actual analyses where this is. It gives you results that are too good in some sense because it's simulated. Like a lot of these simulations, all the variables, if you want to play with it later, are up at the top, right? How many reads get simulated, how many replicates per group. So here, this is going to simulate just 2 different groups, an A group and a B group, and give you FASTQ files for those that we are then going to go ahead and map in the same way, except with different mappers that we did for genome variant analysis. So run that cell. Actually, it's pretty fast to simulate the data. Then you're going to run fastp. So I don't know if you noticed, but along the side here, popped up all the simulated data. So those are our FASTQ files. And we've got, like I said, A group and B group. Why are there 2 files for each replicate? File names tell you. Not yet, because we haven't run FASTP yet. We've installed FASTP, but we haven't run it yet. Right. So, for example, we've got A rep 0 R1, A rep 0 R2. What are those 2 files? I'm asking because it tells you something about the data, the kind of data that you have. Right. So we've, yeah. Yeah, exactly. This tells you that it's simulated paired end reads, right? So you've got your forward and your reverse read from each read pair. And then there's 0, 1, and 2 are the replicate numbers for group A and for group B, okay? So even though, again, this is just click play and the Colab notebook runs, I would like you to think about what it's doing. And then so next cell, just like GVA, it's going to take all those FASTQ files and look at the quality and trim the bad reads. So this is, an example where I said that these data are too good because they're simulated, right? All of the reads pass filter. There are no low quality reads. Okay, you never get that in reality, right? Okay, next step. We'll come back and I'll explain what salmon is. So this is going to do the mapping, pseudo mapping. Which, again, I'll come back to that. But this is all output. We'll come back and look at this. And then this is the step. Step 4 is what takes a minute. So DESeq2, which is the tool that we're going to use to test for differential expression, as of today, is only in R. So I could have switched and used a Python tool instead, but in the literature, you will almost always see DESeq2 used. And this gives me an opportunity to show you how to run an R package in your Python notebook. So this rpy2 is a package that will let us do that. Just installs the R kernel into the background of our notebook. And then way back when I was introducing Colab notebooks, we talked about magic cells. And we've been running bash commands in the notebooks, right? Anything that starts with the exclamation point, it's telling the Colab notebook to run this with bash. But this is this magic cell notation, it says run this whole cell as R, or you can do HTML, whatever. So this is going to use R to go install the Bioconda manager. It's a package manager for R. It's going to go get DESeq2 and install it. TX import. Those are the 3 packages, but it takes a minute. So go ahead and start that, and that's going to spin for a while. And then we'll just put that in the background and go to the lecture. So you should go ahead and download the PowerPoint part 2 slides. The beginning of this, we didn't get very far in the RNA-Seq lecture. We basically ended with mapping to a reference genome. Let's see. So I think we ended here. After this, after I decided to punt the EQTL stuff, gave me more time to talk about the steps in the pipeline, the workflow for the quantification and differential expression. But just reminder quick, so we're all on the same page. What are we doing here? So thinking about the wet lab workflow, we've extracted RNA from our cells, our tissues, whatever, and then talked about how that RNA is going to be full of ribosomal RNAs, and you don't want to waste all your reads sequencing ribosomal RNA. So you will have done some kind of ribosome depletion or poly A capture in order to enrich for the protein coding stuff usually, or could be non-coding, but typically you're going after the coding stuff. Right. And then we talked about some of the advantages of, or some of the things that you get out of RNA-Seq besides just gene expression data. Comes with some cautions, but right, we can get a good idea of intron-exon boundaries because you get reads that split the introns. Talked about allele-specific expression as a caution to whether you can call variants or not, because you can't assume that a heterozygote is going to give you 50-50 allele frequency. Right, the example there is that you've got one read that has that C in it. You don't know if that's a true variant, a base calling error, or really strong allele-specific expression, where it's just, you know, mom's allele has the, mom gave you the G allele, dad gave you the C allele, and there's just really strong bias to express the mom's copy of that gene. So you get way overrepresentation of one allele over the other. So just adds a layer of difficulty to calling variants from RNA-Seq data. You see it in the literature though, so typically you just have to tune the thresholds for what you believe or what you want to call a true variant versus a false positive. Okay, so that's if you have a reference genome, and we talked about there's basically two flavors at the outset of RNA-Seq. Either you're doing that mapping to a known genome that's annotated, or you're assembling the transcriptome de novo, which lots of labs in biology department here work on non-model systems where there's not a good genome that already exists. In that case, you're going to have to do assembly. Really, the only tool that gets used that you will see in the literature is called Trinity, developed by the Broad, and it's called Trinity because it's got 3 steps: inchworm, chrysalis, butterfly. They're really catchy at naming their software packages. I'm not going to go into the details of this. I've given you a video for how they get assembled and then clustered into transcripts. The assembling step, inchworm, and then chrysalis is doing the clustering, and then butterfly is actually producing the transcripts. So there's a video linked in the PowerPoint. That's also the same video. I moved it from Tuesday lecture to today just because it's more relevant for today's material. It's short. It's only 7.5 minutes. It's a high-level overview on how each of those steps works. We're going to talk a little bit about why it's complicated when we get to how you quantify, but Trinity is very computationally expensive. That's why we are not going to do it in class or even assign it as a sort of homework assignment. You would have to do this on Bridges 2, and it would eat up more of our allocation than I think is worth it. Because we talked about how even genome assembly, when we were doing variant analysis, is often a one-off project. You're going to assemble your transcriptome or assemble your genome once and then work on that. The other thing is Trinity is really, it's been around for a while. So if you get it all installed with all the dependencies working, it really is just like a Colab notebook in the sense of you execute it and you get transcripts out at the end. The challenge, like I said, it's computationally expensive. As a rule of thumb, you need a gigabyte of RAM per million reads for the inchworm step. And so if you remember back to Illumina throughput, you could be getting 50, 100 million reads, no problem. And so that's why Bridges has those large mem partitions that have a terabyte of RAM. It's for these assembly steps that are sped up by memory bandwidth. So why is memory the limiting factor? And as a bioinformatician, these are important things to sort of log away to know what's going to limit the speed of my analysis. For assembly jobs where all against all is the rate limiting part of the algorithm, more memory is better, because memory lets you do really fast comparison of read versus read. Okay, if you keep having to go to disk to pull up more reads to compare, that I/O is really slow. Okay, well, comparatively slow. So if you can store all your reads in memory, it can be a much faster job than doing all that read write. And then the complexity of implementing Trinity is that first step is memory limited. And then once you start going to the clustering and transcript assembly, or putting it from the making it go from the clusters to the actual sequence transcripts, I'll show you the output in the next slide. Those are highly parallelizable. So that now you're compute core limited. So if you can spread it out across multiple cores, then that will make it go faster. So oftentimes Trinity takes some specialized setup where you will have it checkpoint after inchworm finishes, give you the inchworm output, and then start a new job that is not on the large mem node. It's on one of the high core nodes. Like the regular memory nodes have lots of cores, so you can split it across. And I have not run it on any of the GPU clusters myself yet, but it has been compiled to run on GPU. So if you can get thousands of cores, this can go much faster. Okay. And then I put in red here that Bridges 2, I think all of the TAC computers, the Texas Compute Center, kill your job after 48 hours. Okay, so if you accidentally start a job without checkpoints in it, and like what I mean by checkpoints, intermediate outputs, where it's writing the results of whatever step it just completed. You could have a job run for 48 hours and then not have anything usable, and it kills the job. So something to be aware of, and a limitation for implementing this on an HPC. Okay, so assuming you can get it running, if you're in my lab, Casto lab, Fujita lab, you have in-house hardware that can run this. The rumor is UTA has a new computer that might let us do it. Don't know. Those are all asides. And the only reason I bring those up is if you decide on a final semester project where you need that compute to do a de novo transcriptome, we can do it even if it won't work on bridges, okay? This is what the output of Trinity looks like. So it's a FASTA file. It's got a complicated header that basically tells you which gene it is and which transcript, or which, how it's related to other transcripts in the assembly. You could use this transcriptome straight off Trinity as the that's your reference transcriptome. That's what you're going to map to. Alternatively, you can just take the longest in the subset. So I put a slide in here just because those names are complicated. So it tells you the cluster identification, which component. So let me skip to the next slide just because I realize there's context here. Transcripts are not genes, right? And this is because I know we have lots of data science people in here that may conflate the two. So why aren't transcripts genes? I think biologists are used to thinking of alternative splicing and multiple exons, but in eukaryotic genomes, my cartoon genome here has a gene with 5 exons in it. The exons are the protein coding part. Various transcriptional regulators will determine which exons get included in any particular transcript. So you can make multiple different variations on this protein because of the exon structure. So the example here, I'm showing one intermediate RNA, or one potential RNA, that has all 5 of those exons in it. But you can have alternative splicing of that RNA so that you get one protein that only has 1, 3, and 5, one that has 2, 3, and 4, one that has 1, 2, and 5. So you can imagine this. That's why, right, these are not genes. These are transcripts. That's the best assembly that you can do from all your 150 base pair Illumina reads. You assemble those. Some of them, right, that's the reality. Some of those transcripts were 135 transcripts, but that's not the gene in the way that you typically think of a gene, because the gene is all 5 of those transcripts. It's just that this one doesn't use all the exons. I just said all 5 of those transcripts, all 5 of those exons. Okay, so hopefully I think that's an important sort of an intuition about some of the problems that arise in RNA-Seq interpretation and sort of this mapping step, assembly step. And so how do you go from this, which gives you a cluster of transcripts that are all related, that's what Trinity's telling you, to something that is more like the genes? The extra layer of problem here is that you could have genes that are completely missing from your transcriptome because you didn't sample all the time points in development, or you didn't sample all the tissues in the organism, right? Not every gene is expressed all the time. So you may have versions of this gene, like transcripts, that are not present in your data set. So just as a thought experiment, right? I only sampled first day larvae of my insect. I don't know, because I don't have a reference genome and with an annotation, that this is a gene that has 5 exons in it. In my Trinity output, I only get the first transcript example here and the last transcript example there. In that situation, I don't even know exon 4 exists, okay? And so later on, when I go to start trying to map, and because now I've sampled day 5 larvae, I get some of these transcripts in there. There may be a reannotation step. There may be like all of those reads may not map because I've got paired ends and one of them doesn't align to what I think is a gene. So all of that missing genes or missing exons is another complication. And then what often happens, so again, this is mostly for the labs or projects where you're working on a non-model system, like how many genes are in a genome is often inflated. Human genome example, the over-under odds, the Vegas odds on how many genes were in the human genome before 2001 when the first reference came out was about 120,000 genes. So that was the expectation. So you go ahead and sequence the genome and you have an expectation of 120,000 genes, and you do some RNA sequencing, it's not high throughput back then, and you get estimates that are in the 40,000 range. And everybody's like, wow, there are only 40,000 genes in the human genome. We were way off. And like this differential splicing, alternative splicing, must be a much more important thing than we thought. Well, fast forward 10 or 15 years, and that number of genes in the genome has been cut in half. There are really only somewhere in the neighborhood of 20,000 protein coding genes that make somewhere on the order of 120,000 different isoforms. So, like, you have to get used to that kind of thinking of the complexity of going from genome to transcriptome. Not always straightforward, and for the most bioinformatics people are employed by clinical situations, or you're working in a mouse or a human lab where this is a little bit less of a concern because you've got a well-annotated genome. So there are 5 exons. You're going to map to that reference. Okay.
How are transcripts added to genomes, like if you sequence a genome, right? So like in the GFF file for the gene, there's so many mRNAs, right? Where did those mRNAs come from?
Gene prediction software or RNA-Seq or both. So I hadn't planned to, well, we're not going to do it for class, but if somebody wants to do genome annotation as a semester project, that would be a cool project that would probably help inform a lot of people. But there's a pipeline called Maker that will take all of the evidence that you can give it. So it will run multiple de novo predictors. So we know there are characteristics of protein coding genes, right? They start with ATG, they have probably a different GC composition than the background. Lots of things that make a particular stretch of sequence look more like a coding sequence. Plus it has an open reading frame that translates amino acids. So all of that stuff feeds into these gene prediction softwares. They'll make a prediction that then puts a line in the GFF file. And to the extent that you have RNA-Seq data, you can also have it use that as reinforcement for the models. And so I've mentioned a couple times a benefit of RNA-Seq is this intron-exon junction, right? So gene prediction software often gets those wrong. Like there's an extra reading frame that it didn't include in the gene model. RNA-Seq will tell you for sure very precisely which nucleotide ends the reading frame and starts the intron. Okay. So all that is to get to a transcriptome. Trinity has built-in software or built-in packages to help you go from that raw transcript clustering to something that's more like genes. You can tell it to only include the longest isoform, or you can say just keep everything, or you can tell it to collapse everything that's 95% similar. Various tools in there that will try to help you go from those transcripts to genes. Just a word on best practice. If you're doing this de novo type assembly and sequence expression quantification, you want to put all of your sample data. So in our cartoon example, everything from group A and everything from group B in one big pool of data and use that big pool to make the transcriptome. The reason for that is that you'll get the most comprehensive set of transcripts that way. So that's becoming your reference, and then you're going to take your individual samples and remap it to that comprehensive, quote unquote, transcriptome. Okay. How do you know if it's any good at the end of the day? So the tool is called BUSCO, stands for Benchmarking Universal Single Copy Orthologs. This is a very well curated set of genes that we know exist in single copy across everything where we've got well annotated genomes. And that grows, weekly. What it will do is take your transcriptome, take the gene models for all of these single copy universal orthologs, and say how many of your transcripts are in that set. Okay? And so you can imagine if you have limited developmental time or limited tissue sampling, your transcriptome is probably not going to be very complete because you haven't sampled all the tissues or all the time points. But BUSCO will give you an indicator for how complete is your transcriptome. And so this graph over here on the right, this one isn't testing different species, it's testing different assemblers. And the x-axis here is the percent of those benchmarking orthologs that were in the transcriptome. And so the way this is color-coded, the red is missing. So you might argue that the Velvet Oasis K25 assembler, doesn't really matter to know the difference between the assemblers, but the graph is telling you that's the worst one. Like it's missing the most. And all the data was the same going into this experiment that's being shown. So this was to test how good is the assembler. The light blue are those complete single copy orthologs. The dark blue are complete and duplicated. And then you've got a yellow chunk that are fragmented. The duplicated and fragmented ones, those could be real, right? It could be pseudogenes because we're looking across species, right? And there could be duplicate genes in there. There's not a sort of computational way to know without really going back. Actually, anybody have an intuition about how you might look? Like, say BUSCO tells you one of your transcripts or genes is duplicated. It's finding two copies of what otherwise should be a single copy universal ortholog.
Are you saying computationally or?
Yeah, computationally. How might you check? This is trying to wake you up because I know I've been talking for a while. Think about how they're laid out in the genome. The question is it single copy or is it duplicated? No guesses? Oh, how about this? How would you know if one is missing? Right, so in this red category, what that means is it's got a gene in the database of single copy orthologs, but they don't find it in your transcriptome. Let's pretend we also have some genome sequence to go with it. How might you go look to see if that gene actually exists in your genome? Yeah, blast it, map it, whatever terminology you want to use. Yeah. All right, so you take the gene model from the BUSCO database, blast it against your genome, and ask if it's there. Okay, it's not in my transcriptome, but it is in my genome. Tells you that just wasn't expressed in whatever tissue I sampled. Okay, so back to the duplicate question. Like, how might you ask if this is actually duplicated, or I just have two transcripts that should be collapsed because they're actually only one gene in the genome? Okay, the answer is synteny. What does that mean? Same orientation in the genome sense. Okay, so you can look to see if does this transcript actually occur twice in my genome. And so I talked briefly last time about how gene family analysis sort of probably got me this job in the first place, and how bad the chimp genome was when we first started working on it. We had to do a ton of, like, we couldn't get the stuff published because nobody believed us because the chimp genome annotation was so bad. So we had to go back and look when we got results like this to prove that that gene was actually missing in the chimp genome, or this gene was actually duplicated in the human genome by looking at, okay, this stretch of human genome actually has two copies, and if you go look in the chimp genome, there's only one copy in that region. Okay, that's like the evidence for absence or the evidence for duplication, right? It's just the two sides of the same coin. Okay, so that's what you kind of have to do if you get messy transcriptomes. Okay, but ideally, you know, if you've got 95-ish%, which if you've sampled well, you know, enough developmental or enough tissue time points, totally common to see 90+% of transcripts in a BUSCO analysis. Okay, that's assembly and Trinity. That was a long time on something that an approach that we're not really going to use. But hopefully somebody, and I know lots of labs in the department are doing it. So let's move to mapping and quantification. So we've got gene models now. So looking at our overall workflow / pipeline, you need the gene models in order to go map. And then you're also going to need the gene models, you know, to do the quantification. So even if you have good gene models, and from here on out, I'm going to sort of assume we're working on something that's like the human genome, where it's got a good reference genome, well assembled, and good annotations for where the genes are. Like, we know the intron-exon boundaries. But there are more things to consider. So how do we calculate the expression of a gene based on the read counts? So this is a cartoon example where we've got a blue gene and a pink gene, and each of those little black bars is a read that mapped to that exon, right? So this blue gene has 3 exons, this pink gene has 3 exons. Those 2 genes are the same size, and so we can just count up the number of reads, and that is our proxy for the expression level, right? In our soup of RNA that we extracted, if you've got more copies of that RNA, you're going to get more reads from that kind of RNA. So this, if the genes were all the same size, that would be easy. Our read counts would be basically a one-to-one association with our expression value. The problem is genes are not all the same size. So even if we think about the same genes, but you're using different exons in that gene. So the green gene's got 2 short exons. 4 has 2 exons and much larger. So here we've got way more reads. If you were to count up the number of black bars, you would see there are a lot more reads that mapped to the orange gene than to the green gene. But that's just because there was more of that RNA molecule to grab reads from. Does that make sense? If you've got more of the molecules in your soup, you're more likely to get a read from that molecule. So if the gene is bigger, you're going to oversample it just because it has more of the constituency of the soup. Okay? So we have to normalize for that. The way you do that, you often see RPKM or FPKM. They're analogous. Reads per kilobase million. This is controlling for the size of the gene and how much sequence did you get. Okay? Because the other thing is, if you sequence individual 1 and get 50 million reads from that individual, in individual 2, something goes wrong with the sequencer and you only get 30 million good reads, right? Because we're going to run FastP, and right, sequencer just did a worse job calling bases on the second run. If there's 50 million from this individual and 30 million from this individual, it's going to look like all the genes in the 50 million are expressed higher, right? Because you just have more reads. So you have to both normalize for how many reads did you get and normalize for how big was the gene. So an increasingly common one is TPM. Not going to work through this, and you've got the slide. Also given you an RNA-Seq blog post there so you can go read. But hopefully, if you get that level of intuition, I need to account for how many reads were in my library and how big was the gene. That's really all you need to know in terms of the intuition for how the normalization works. Because the software is going to do the actual normalization for you. You don't have to write code to do that normalization. The sort of final issue that you have that goes along with alternative splicing is that here I've got an example, 2 isoforms from the same gene, right? And all the blue chunks are exons that are used in both isoforms. And the red and the yellow are exons that only get used in one of the isoforms. And so you run into this question when you're mapping the reads of, well, did these blue ones come from isoform A or isoform B? Because when I now infer the expression level of this gene, that's going to matter. So many of the sort of modern mappers, and we're going to use salmon. These are just some of the ones that do a good job of this. They use an expectation maximization algorithm, and all that does is sort of say, what's the average depth of the unique parts, and randomly associate the duplicate mappers to one or the other, so that the average read depth is the same as the unique parts. So what this is doing, the cartoon is meant to show, is you've sort of taken what's the average of the red depth and the yellow depth and made the blue depth be about the same as the red and the yellow. Does that make some sense, I hope? It was something that tripped up early mappers a lot because, right, this one is sort of, okay, you might say no problem with that one, but the average expression level of isoform B you might infer to be lower than it should be. Or actually, I should say higher than it should be, because you've got all these duplicate mapped reads that didn't actually belong with that isoform. Took us a long time to figure out what was going on with that. Anyway, which mapping tool to use? Right, the sort of practical question of how do I make this run? There are three main ones that get used a lot nowadays: STAR, HiSat, and Salmon. STAR and HiSat are similar. They are true aligners, where it's taking the reads and then doing the process like BLAST, where it seeds and then extends to find out where in the genome does this map best, or where in the transcriptome does it map best. I put in blue the sort of key things to consider when you're thinking about which one should I use for my data if you're doing this for a project or in the lab. STAR is really fast, but it's a memory hog, okay? And so you have to be on a machine that has a lot of memory available if you're going to use STAR. HiSat2 is not as fast as STAR, but it's better on memory usage. So if you've got a reference genome, and especially if you want to know intron-exon boundaries and stuff like that, you might want to pick STAR or HiSat, and then the decision between those two is your computational resource. You know, how much memory do I have and how much time do I have? The outputs for STAR and HiSat are essentially the same. They're going to give you a BAM file. Just like our BAM files from genome variant analysis, this is just it's that shorthand form of where do the reads map in the genome. Salmon is a little different. It's a pseudo-aligner. What pseudo-aligners do is they take only the transcripts. So even if you have a reference genome, it will pull out only the coding parts. And it breaks up both the transcriptome and the reads into k-mers and does a similarity matrix and just counts up the similarity. So it's not doing true alignment. It's just counting how many of my reads from each k-mer are associated with each k-mer that came from each transcript. Turns out that is super fast and doesn't require very much memory. So speed and memory use are, and it also turns out to be very accurate, okay, in terms of quantification. The potential downsides is you're not mapping to the genome, so it's not going to give you any information about intron-exon boundaries or non-coding RNAs, because those are not included either unless you explicitly put them in. And it's not going to give you a BAM file because it's not doing alignment in the classical sense of, you know, finding out where in the genome does it fit best. So the output from Salmon are quant files, which can either be TPM, right, the normalized version, or the raw counts. And I point out the fact that you can get raw counts because that's what you need for DESeq. So that's a very practical, don't feed TPM into DESeq. In fact, DESeq might choke if you try to give it TPM. So that's mapping and quantification. Oh, I skipped over quantification for the top two, right? You get a BAM file as output. That's not quantified gene expression. You've got to use another tool. Feature count is the name of the tool that's typically used in the STAR or HiSat aligner. There's also HT-seq count. How many of you have used Galaxy already for, have you done RNA-Seq on Galaxy? Yeah, so probably you use star or HiSat to do your mapping, and then you've got to use feature count or HT-seq count as a downstream, right? So you've got to do a mapper and then a counter, so that the mapping and quantification steps are separate if you use one of those mappers. The reason I've sort of glossed over that counting step is it's just like the variant analysis where it does the pileup and just counts the read depth, okay, and then takes an average. Salmon's is a little different though, because it's doing the quantification at the same time it's doing the mapping. You can give Salmon a BAM file. So if you've already done your mapping, you can skip the mapping step and just have Salmon do the counting. And then differential expression, the reason we started that pipeline in the background is DESeq2, which is what everyone uses, only has an R implementation right now. Last major, well, I don't know, this is sort of a design and analysis issue of how deep should I sequence? So, like, just to make sure you're tracking with me, what do I mean by sequencing depth? Yeah, how many reads there are, and what determines the sequencing depth, very broadly. In RNA-Seq, how often it's transcribed, and then like the average sequencing depth across all transcripts is determined by how much money did I spend to get more reads. I gave the analogy earlier, 50 million reads versus 30 million reads. If you spend more money to get more reads, you will detect more genes, right? Why is that? Like, somebody connect the dots for me here. Why will I detect more genes if I get more total reads? Yes, at the end of the day, that's true. Why? Like, what genes am I picking up? That, like, if I sequenced at 30,000 or 30 million, and I now go back and sequence at 50 million, which genes are being added to that? May not be asking this question very well, but that I'm picking up by sequencing more. Remember, think back to our quantification, okay? There are more copies of that messenger RNA, I get more reads from it. So now we're talking about the ones that I didn't detect because I didn't have enough reads, right? Those are the ones where there's not very much messenger RNA in there. So, answering my own question, the lowly expressed genes. So the more sequence you gather, the more power you have to detect genes that are sort of barely on. There are very few transcripts in there, okay? I love to play board games. A mechanism in board games is bag building. Like, how many of a certain color bead are in the bag determines my probability of pulling one out. If I get to draw more times, I'm more likely to pull out that bead that there's only one copy of it in the bag, right? That's what's happening with read depth, okay? Think of reads as how many times do I pull out of that bag, okay? And your chance to detect the rare ones are increased if you sequence more. But that raises another potential issue of do I really care about the rare stuff? Okay, like we know that there is spurious transcription, that it's not really biologically meaningful, and because it is random draws, the lowly expressed stuff, even if I sequence both of these things at 50 million reads, I'm randomly going to sample from the low expression stuff. So on that low expression stuff, do I want to say, oh, this sample is expressing that gene, but this one isn't? Okay? No, it's just that you randomly sampled that one. So that all goes to the statistical power, which is what we're trying to get to, because the biological question behind all of this is, are the genes differentially expressed between two individuals, two treatments, whatever? And so, right, putting some numbers on that in case it's not intuitive yet, how confident in that biological interpretation for gene A to say that that is a significantly differentially expressed gene? Because gene A and gene B, they have the same fold change. Like, is gene A expressed twice as high as gene B? How many are confident that gene A is expressed twice as much as gene B? Oh, good. You all pass. Whoops, how come it didn't? Oh, I know why, right? So you would not want to imply, and so when we think about how our statistics need to work, you don't want to say that gene A is significantly different, but you do want to say probably that gene B is different. And this is also the first indicator for why fold change does not tell you about significantly different expression. Okay, so just because something is, inferred to be twice as high, you got twice as many reads, even twice as many normalized reads, that doesn't necessarily mean it's significant. It depends on how much noise there is in your data, right? And so just if you were doing a Fish's exact test, which is one test you might do on these examples, right? Again, just trying to drive home the point that more reads is more statistical power. Okay, so I'll finish these two slides and then we'll go back to the Colab notebook. So DESeq, what is it doing? Like, why not just do, say, a t-test or an ANOVA to see if our gene expression level is different between our treatments? Right, so in our practice simulated data, we've got A group and a B group. Why not just do a t-test to see if the average of the A group is bigger than the average of the B group? Man. Well, because of the assumptions of t-tests, and they don't fit with the distribution of RNA-Seq data, right? So some of those assumptions, some of the reasons, like integer counts, they're not normally distributed because you have to account for the depth and the gene length, because they're highly variable across replicates. Like, what I mean by highly variable, they are over dispersed. Talk about, so that's where the negative binomial distribution comes in. That's negative binomial distribution. Going down a rabbit hole that I didn't intend to, but now I'm realizing it. It might be important to think about, because I know a lot of you are in biometry right now too. So, right, if you're doing a t-test or an ANOVA, you make some assumption about the distributions of the underlying variable, right? And so what those tests are doing is saying how much overlap between those distributions before I say this is significantly different. the reason the assumptions of those tests are important is that if the distributions are not normal, like this one is like this and this one is normal, now the sort of test is not valid because the expected distributions won't overlap in the same way. Okay, so that's why DESeq understands that the count data is not normally distributed. So it uses a negative binomial as an assumption, and negative binomial says that for a given mean, you expect the variance to be much bigger than you would otherwise get in a normal or even a Poisson distribution, which assumes that the mean and the variance are the same. Do you have a question? Yeah.
Yeah, what I'm curious about is, like, up until this point, I've always been told that it's not going to be super common with biological data to just run a t-test. So you do a non-parametric or do a transformation. So for DESeq2, anytime you have like DNA type biological data, is it always better than to just do this and not do like a non-parametric t-test or transform the data?
Yes, because DESeq is also going to account for gene length and all those other things that we've talked about that are particular to RNA sequencing. Yeah. Okay, so these are the steps for DESeq, and I wanted you to have like what's going on because you're just going to click play in the Colab notebook and it's going to generate it's going to tell you that they are or are not, the genes are or are not differentially expressed. And this goes to your question as well, like that normalization that we talked about for the size of the library and the size of the gene, that's the first step. It's going to estimate the variance, so it has to do that in order to do step 3, where it sort of pulls in the extreme outliers. Okay, and this is a step that you sort of wouldn't have to do, but we know that because of technical noise and the biological noise, like gene expression is a very noisy trait. Okay, that's an observation. Like one cell to the next, one tissue to the next, one time point to the next, and then so that level of noise plus the random sampling of by the reads, what I was talking about, the bag pulling, that adds noise. That combination of things adding variance just has been shown to be improved in the testing if you use a denoising step, essentially. And yeah. Then the next one is just to run a generalized linear model. It assumes that negative binomial. And then we're going to talk about multiple testing corrections, but DESeq2 outputs the false discovery rate as an adjusted p-value. So you can read the adjusted p-value from DESeq output as like a 0.05 adjusted p is FDR 0.05, the false discovery rate 0.05. And I'll unpack all of that if that was total stats jargon to you next week. The last thing in the Colab notebook are going to be 2 plots like this. Volcano plot, which the x-axis is the fold change, and the y-axis is the p-value, or negative log p in this case, just so it scales where more significant is higher. And so oftentimes when you do differential expression analysis, you're interested not only in the statistical significance, but the fold change. Because I talked before about how fold change of 2 might not be statistically significant. If you have really good sampling and there's not very much noise in the gene expression, you can have things that are statistically significant but have very low fold change. And so you also have the question there about, is that biologically relevant, right? My fold change is 1.2. So they're barely any different, but my stats are so good, I've got a big enough sample that it's still statistically significant. I don't know if I want to interpret that biologically. And so this volcano plots are meant to try to show both of those things at the same time. So the things that are color-coded here are the things that are both significant and they're like, so their negative log p is 2 point something. I don't know what happened to my cursor. I can't draw. Oh, it's over there, but it's not on my screen. There we go. Right. So these are things where the fold change, this is log 2 fold change, so log 2 of 1, that'd be a 2-fold change. So if you're to the left of that, and now your p-value, that's probably 0.05. I don't remember off the top of my head the translation. So those are both significant and have a fold change greater than 2. That's what the volcano plots are meant to show you. And then over here, these are heat maps. So typically the way these are drawn and the way the Colab notebook will draw them is that the columns are your samples. So, right, we've got 3 A samples and 3 B samples. So we've got, that's what it's showing at the top. And ideally what you're going to see is like the 3 A samples cluster together, the 3 B samples cluster together, and then the rows are the genes. And so it's showing whether compared to all other genes, this is not all other genes, all other samples within that gene, is it upregulated or downregulated compared to the average across all the samples? Right. So we'll come back to that in our Colab notebook in a second. And then the last slide in this is just the elephant in the room when you talk about RNA-Seq data. The biological question is what? What's our biological question for RNA-Seq? Just keeping you awake, not insulting your intelligence. Why are we doing this experiment? Yes, exactly. And so the temptation, right, that's exactly what this analysis is going to tell you. Relative gene transcription, okay? So the thing that is often implied and is that transcription has a strong correlation with translation. So those transcripts are getting turned into protein, and then that has the functional consequence, okay? For protein coding genes, it's the proteins that actually carry out the functions, and we're only measuring transcription. The elephant in the room is that protein quantification is still a really hard thing to do, and we're going to just assume that our transcripts have a sort of one-to-one correlation with protein. And so differential transcript abundance has a one-to-one correlation with sort of functional difference. And that might not be true. And there's been studies to show that it is absolutely not true. So there's a whole nother layer of gene regulation here, the post-transcriptional gene regulation that we're sort of ignoring in RNA-Seq. RNA-Seq is super popular, probably, for people who are doing the genetics of phenotype, one of the most common experimental designs or experiments you might want to do. That's really because it's cheap and easy, not because it actually tells you the answer to functional, right? Now, that's the gloom and doom. That's the elephant in the room that nobody talks about. So it's good to have that caveat in mind, especially when your transcription analysis, your RNA-Seq doesn't make biological sense. Maybe there's another layer of regulation going on. but there's also lots of good evidence that there is strong correlation. Lots and lots of studies that where the RNA-Seq analysis makes very good sense with the phenotype. So something to be aware of, not to be paranoid about. Okay, let's go back to our notebook. Surely by this time your R packages have been installed. And I went back and what's going on in this cell, this is running R. So if you don't recognize, well, the top part is bash. That's why the exclamation points are there. But if you don't recognize that from what we know about Python, that's because it's not in Python, right? So here, this samples with the arrow going to the left, that's the assignment in R, where in Python you just say equals. So one of the many reasons I like Python. So I've gone back and added lots of comments so that you can see what each of the lines are doing here. But this chunk of code is, well, it first installed those R packages, then it installs DESeq and TX-import, then it uses TX-import to take the salmon quants. So over on the left, remember we had the FASTQ files. You'll notice that your runtime has been populated by these quant directories. In those quant directories, if you dig, you'll find the quant SF files. That has the expression level in terms of read count or TPM for each of the samples. So that's the output from salmon. So our mapping and our quantification got done in one step by salmon. This R cell is taking those quant files, converting, and actually it's taking the not the TPM, but the raw counts, and feeding that into DESeq. Okay, so that you don't get down to that until line 57. Creates a DESeq2 object. What that's doing is saying for the DESeq object, these are the data, and this is the experimental design. So that's something that we're going to gloss over for today, but the slides have links. Like what happens if you've actually got 3 treatments, or you want to do all the treatments compared to a reference. Like you've got 2 mutants and you want to compare it to something you know is wild type. Okay, that's where you would be specifying that experimental design so that DESeq knows which comparisons you want to make for differential expression. Okay, we're doing a simple, we've only got 2 treatments. It's just going to do A relative to B. Is it up or down? Okay, so the output of that cell, okay, that's what, if you were running DESeq in Galaxy or on its own in an RStudio session, you would see the same thing. It's not actually telling you which genes are up and down regulated, but it is generating the, actually, did it finish? Yeah. Actually, I'll show you because this will read that file. So do the create volcano plot. And so it goes and takes the output from DESeq2. This is just a graphical version of the DESeq2 output table, where it's read the adjusted p-value column and the fold change column and plotted all those values for each gene. I flipped the red and the green just because, I don't know, felt like green should be up and red should be down. But again, this is simulated data, so there's not much story to talk about here. The genes have numbers, not names. And then the last cell there, we'll create a heat map. This one's not sorted as pretty as the one that I had in the slides. So the rows, there's no dendrogram, there's no tree because the ordering is arbitrary. But the code to order them, to cluster them, is actually in this cell, right? You could change this to true. Whoops. And this is the same heat map, but now it's done clustering just to organize it so that the up and down regulated genes show up closer together in the figure. And the reason I point that out is there's some functional interpretation here. Now, this one has an equal number. It's simulated data. You can kind of see that from the symmetry in this graphic, right? You've got about the same number of up and down. But when we look at real data, it won't be quite so symmetrical. But, you know, going back to thinking about biologically what's going on, right? It seems like maybe there are a whole bunch of co-regulated genes, right? So the A group has a bunch of genes here that are red. That's upregulated stuff. And then those genes are relatively downregulated in group B, and then vice versa for this group of genes over here, okay? And so whether that's heat tolerance or something like that, you might, right, there's a pathway of genes there that are being co-regulated. That's the inference, okay? We will get more into that interpretation on Tuesday when we look at how do you marry this output with the genome variant stuff that we talked about. Like take each one of these rows as a phenotype and then look at what are the genetic variants that determine that up and down regulation, or are there genetic variants that determine the up and down regulation, okay? So the assignment will just be to, and I'll put them in the instructions in Canvas just like I did last week, to give me those figures as output, just to make sure that everybody's run the RNA-Seq pipeline. Questions before we go?
Do we need to turn it in how it is, or do you need us to do the separation?
It's going to be the same deal where I want your name and ID number as titles instead of the default titles. No, you'll have to add a title. Yep. The pre-clustered one? No, do the clustered one. Yeah.
When you say do the cluster, do you mean how you changed it so that the categories were more separated?
Yep. So the cluster rows, you change that from false to true. Okay, thank you. Again, I said this early on, but you can play with the simulation. Like, again, it's kind of, this is the pedagogy thing here of I gave you a Colab that does it all for you. You could learn nothing if you decide to. The more you play with that Colab notebook, the more you will learn, okay? Yeah. Yeah, for clustering.